And this teaches us several really important things. The first thing it teaches us is that these bioelectrical patterns are in fact instructive. It's not just that, oh, I messed up the pattern and now the face is screwed up. It's not that. It's that you can actually induce very targeted, coherent changes. So we know it's instructive. It carries information, make an eye here. That's the first thing. The second thing is the pattern that we put in is extremely simple. It doesn't have all the information that you would need in order to build an eye from scratch. If somebody wanted to really, let's say, 3D print an eye, I mean, nobody's done that. Not even sure it's possible. But if somebody wanted to do that, you would have to have an incredible amount of information. We didn't pass all that information on.
What generates that pattern? And if it is the cells or the proteins, where in the protein is that memory encoded?
So this is the more difficult question, and I think we need to really make progress on this. We really have to redefine what we mean by where.
Hello, everybody, and welcome back to Chasing Consciousness. So today we have the extraordinary role of bioelectricity in the orchestration and elaboration of organisms and life to look at. I became interested in this in the 1990s when I read a book that was controversial at the time called The Body Electric by Dr. Robert Becker, who'd been studying the bioelectric fields around salamanders as they regenerated their limbs. And I thought this research had died out. I've been looking for it for years. And that was until my guest, Zhen Xu, at the University of Michigan, spoke about the work of my guest today in our episode number 37 on her work, Histotripsy, which is ultrasound for destroying cancer cells. He is none other than the award-winning biology professor at Tufts, Michael Levin. He's in the Department of Regenerative and Developmental Biology, although he started out as a computer engineer. His specializations are in how cells form these bioelectrical networks for storing and recalling the pattern memories that guide morphogenesis. He then applies that to next generation AI to help us understand this top-down control of pattern regulation in the new bioinformatics of shape. He is a complete visionary in how all this can then be applied to regenerative medicine and bioengineering, and his work really forces us to examine our approach to morphogenesis. Now, I've been longing to find someone to talk to about the implications of this work for the bioelectric nature of collective intelligence, in this case of cells, how that builds up at ever higher levels and layers of collective cellular agency, cognition, sense of self, culminating perhaps in collective intelligences greater than single organisms. But for the answer to that all-important question, we're going to have to wait and see what Michael says. So without further ado, let's go. Michael Levin, welcome to Chasing Consciousness. Thank you so much for coming on the show. How are you today?
Great. Thank you so much. Happy to be here.
Well, thank you so much for giving us your time, Michael. I know how much you're in demand these days. Now, I love to start asking my guests about their earliest deep questions and reflections about life, about the world, usually sometime in late childhood, early adolescence. What can you remember from that time that may have left some trace that persevered into your adult life work?
Yeah, I remember two general things. One is that my father used to take the back off of the TV set that we had. So we had this like giant TV, this big wooden box with a little tiny screen in front and vacuum tubes. I mean, most of the people in this audience have no idea what that looks like, but it's this thing with glowing vacuum tubes in the back. And so he would take the back off and we would just sit there and look at it. And I remember just being just in total awe of how did somebody know to put all the pieces in exactly the right way? Because it's pretty obvious it wasn't random. How did somebody know to put all those pieces in, right? And so that I think that was the beginning of my love for engineering and technology and computation and things like that. And on the flip side, I had a friend who was a little older. So this was, I must have been 5 or 6 years old at this time. And I had a friend who was a little older, and we would go outside, and he was really into insects and collecting bugs and insects and caterpillars and things. And so he would show me all this stuff. And that was kind of the other end of the spectrum. And I would look at these things and I'd say, wow, yeah, they also have parts, but the parts seem different. They seem to have preferences about what happens next, which the TV doesn't seem to have. And then, so what's the same, what's different, right? And so that's it. And just watching eggs in particular, various insect eggs and the larvae that come out of it, and then they turn into an insect, and just watching that metamorphosis of a living being coming from almost nothing. From a little blob of inanimate chemistry. That's I think that's it. I sort of put everything back on that.
Yeah, and it's funny how those two things link in your work, both the technological side and the bioengineering side, and we're going to get right into that, listeners. So before we get on to the implications of your work for the formation of cognition, of self, of agency, let's get straight into the meat of today's conversation, the data about bioelectricity and its role in orchestrating new organisms. Now, most of us in the public assume that DNA and the genome are responsible for the form that our bodies generate as we grow and which cells become which organs and tissues. But over 100 years of research, not well-known research, into bioelectricity's role in the new life, and your research on it in growing tadpole embryos has shown that DNA is really only half the picture. Now, many biologists are still assimilating all of this into the consensus. Can you tell us what the results of your bioelectric field manipulation experiments in these tadpoles were, and how it's just changing the way we see the purposing of new cells as different body parts are generated in morphogenesis?
Yeah, and we've done this work in numerous model systems. So we've done planaria, which we can talk about, flatworms and frog, and the human cells as well. But let me back into this a little bit, because I think there's a more profound aspect to this, which I think is even harder for people to get their minds around than simply this idea that, okay, we've had chemical signaling and we've had biomechanical signaling, and eventually people will come to grips with also including bioelectrical signaling. I think this is a more there's a deeper issue here that I want to back into as follows. If you think about what we are, you and I and everybody who's listening to this, in one sense, we are a walking collection of cells, neurons, various other cell types. But we know that there's something more than this, because we have goals, preferences, and memories that don't belong to any of the individual cells. So you can think about very simply, if you take a rat and you train the rat that if he presses a lever, he gets a delicious reward. But there's no individual cell that had both experiences, right? The cells at the bottom of the foot got to touch the lever. The cells in the intestine got the delicious reward. So who owns this associative memory that these two things are connected? Well, there's this thing called the rat, which is actually a very non-trivial kind of thing to find, because if you take a kind of a reductionist approach, you don't see a rat, you see a collection of cells. In fact, you don't even see that, you see a collection of chemicals. And so you have to ask yourself, what is this amazing cognitive glue that binds individual components, in this case cells? Let's just stick with cells, although I can tell a very similar story about molecules, but let's say just cells. And what it does is it binds these cells to a common purpose that exists in a space that these cells don't know anything about. So, right. So these memories of, for example, associative conditioning of the pressing and the pellets and so on, and all the other things that the rat wants to do, and all the things that you and I want to do, they exist in various action spaces that your individual neurons don't know anything about. So you need, right from the get-go, you need some sort of a cognitive glue, a mechanism for binding individual pieces together into a coherent, emergent self that does things that these individual pieces don't do. Now let's look at embryonic development. We all start life as a little blob of chemistry and physics, so a little unfertilized oocyte, a quiescent oocyte. And before you know it, very slowly, that develops into some kind of amazing animal organism, for example, or even something like a human, maybe even a philosopher who will then say that they are not just a mere machine and that they do new things and so on. But it's really critical to take developmental biology seriously. For that reason, actually, I think developmental biology is kind of the queen of all sciences, I think, because there you see in front of your eyes, in the case of frogs and other organisms, that you see it right on the table in front of you, how mind and matter relate. Because you start with something that people think is well described by chemistry and physics, and you end with a complex metacognitive creature, let's in the case of a human. So that process, first of all, so two critical things about that process. One is that it's slow and continuous. There is no special point at which just physics becomes cognition. The developmental biology offers no support for any kind of magic red line there. So it has to be a continuous, slow and continuous development. And the other thing is that it is also very much a collective intelligence problem, because that process is not hardwired, meaning that if you come in and introduce various changes, you move things around, or early embryos, you can cut them into pieces. The cells will do what they need to do to get to where they're going, which is this journey in this space of possible anatomical configurations. They start as an egg, they have to get to whatever. You can deviate them, and they will still get to where they need to go within limits. Of course, you can push them past that limit. But so this matches William James's definition of what an intelligence is. It's the same end by different means. It's the ability to adjust to perturbations and get your goals met. So these anatomical problems that these things solve, and there are many cool examples of it, but these anatomical problems that they solve are also being solved by a collective intelligence. Because, for example, in the case of a salamander, you cut off the limb, it will regrow a new one. No matter where you cut, it'll grow exactly what you need. No individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely does, because you can see it will reach that goal and then stop. So again, you have this parallel idea, right? Of we had a collective of neurons solving problems in three-dimensional space, and that's classic cognition. And now you have a collective of cells solving problems in anatomical space. Those two things, I think, are profoundly related, and both of them call for the same thing. There needs to be a cognitive glue. How is it that individual cells are bound together to common purpose, a purpose in a space that they don't know anything about? So it turns out, and if we take evolution seriously, then maybe it's not such a shock. It turns out that the mechanism in both cases is exactly the same. It's bioelectricity. In the brain, we already know this. In the brain, neuroscientists will already tell you that the thing that binds neurons together to form a unified collective intelligence is electrophysiology. It's the network of neurons and other cells. Turns out that trick, evolution found that trick way before brains actually showed up. That trick was discovered around the time of bacterial biofilms. And there's a collaborator that I have, Gorol Suel, who does this amazing work on bioelectrics, pulling together individual bacteria into a coherent biofilm. This was already here, evolution already discovered it. And since then, you can ask, before we had brains and muscles and could move around, what were these networks thinking about? Well, they were thinking about shape and anatomy. That's what they were thinking about. And so this is the work that we've been doing, is trying to appropriate a whole bunch of concepts from neuroscience, which I don't think really has anything to do with neurons. I think neuroscience is really about multi-scale information processing and scaling of cognition from very humble, little, tiny kinds of agents all the way up through very impressive ones. And we try to take all those tools and use them to understand morphogenesis. So morphogenesis, the collective activity of cells that build and repair and remodel specific anatomies. And so once you understand this, then it much like with neuroscience, and in fact, much like people have done with by training dogs and horses thousands of years before we knew any neurosciences. Why could we do that? It's because all of these things offer this amazing interface. They offer this amazing bioelectrical interface, meaning that the surface of the cell is covered with ion channels and gap junctions. Gap junctions are these little electrical conduits that let one cell signal to another cell electrically. And ion channels are basically little tiny batteries that provide a voltage gradient a voltage difference across the membrane. So once you know that interface is there, you can take on the task of trying to decode well, what is the collective thinking about? What are the patterns there, right? And then to extract that information and then to write it back in a new form. So can we incept false memories into the collective intelligence of cells? And that's what we've been doing. And we developed the first, around 2000, we developed the first molecular tools to read and write electrical information outside the brain. And that's what we've been doing. And when you do that, we have applications in three broad areas. So birth defects and repairing birth defects, inducing regeneration, so the rebuilding of novel structures, and normalizing cancer. Those are the big areas that we've been addressing.
And so much of that we're going to get onto in just a moment, Michael. And I just wanted to start with something that the listeners can really visualize. Just describe how the tadpoles, that are such a perfect way to study this because they're so different from their adult selves, describe how the bioelectric field would be used to choose to know where to place these various organs, eyes, mouth, etc., and what happened when you manipulated that field, because I think that'll really give the visual example that the listeners need.
Yeah. So there's a simple way to describe this, and I'll give you the simple version. But let's just keep in mind there's a very subtle piece of this that we do not really understand very well, either in the brain or in morphogenesis, which is fundamentally how cognitive propositions, so memories, goals, and so on, are encoded in this electrical medium. So I can tell you about a few examples that we've actually cracked and uncovered, but this is probably just a few % of what's actually out there. So I want to be upfront right from the beginning that it's not that I don't want to tell you the whole story. Nobody knows the whole story, and they don't know it for the brain either, and it's exactly the same problem, I think. But anyway, but here's what we do know. What we do know is that if you track so what we developed were voltage sensitive fluorescent dye. So these are chemicals. The chemicals existed. We didn't develop the chemicals. But what we developed is the use of these chemicals to bathe an embryo, an organ, whatever. So bathe some sort of system, and these molecules will fluoresce. They will give off light in different amounts depending on their local electric field across the membrane. So what you can do then is, for the first time, because it used to be if you wanted to know the voltage across some cell, you would have to poke it with an electrode, and cells you might imagine are very unhappy about that. And plus, you've only got the voltage of one cell, so that's hard. So this allows you to look at the whole thing, and we literally have not only pictures but videos, so you can actually see a time-lapse video of, let's say, a frog embryo developing. Danny Adams in my group made the first voltage movie of an embryo developing. It's an absolutely kind of a mind-boggling to see that for the first time. You're literally peeking into the mind of that collective intelligence, making decisions about where the head, the tail, the left, right, dorsal, ventral, where all these things are going to be. So I'll give you a couple of simple examples. So one example is you look at an early embryo that's trying to put its face together. And what you find is that before all of the genes get turned on that build the different structures of the face, there's a bioelectric pattern. And that bioelectric pattern, we're very fortunate because most patterns aren't like that. But in that particular pattern, it's pretty easy to decode. Why? Because it looks like a face. We called it an electric face the moment we saw it because you can immediately see what it's going to be. Here are two spots where the eyes are going to go. There's some placodes out to the side. There's the mouth. You can see exactly what it is. It's one of the rare times when the memory trace actually looks like the thing that it's supposed to be. So you look at this thing and you say, okay, if this is really instructive, because that's the real question, right? Is this pattern that we're seeing, is it instructive for what happens next? So you might say that if it's instructive, what we ought to be able to do is recapitulate that pattern somewhere else. So what we did was, and we took so there's a particular spot of voltage there that says build an eye here. That's where the normal eye shows up. So to prove that, what you do is you take some RNA that encodes these ion channels. So it's basically a molecule that when it gets into the cell, tells the cell to build a particular kind of little voltage element that's going to sit in the membrane and give you a certain kind of voltage. So we make some RNA for these potassium channels. We chose them specifically because they would create a voltage that will be like what the eye spot is, and we inject it into a region of the embryo that will be something else outside the head. So gut, tail, whatever, somewhere else. And sure enough, those cells get the message. The voltage pattern arises, and they will create an eye. And that eye has all the right components, all the right stuff inside. In fact, the animals can see out of ectopic eyes. And this teaches us several really important things. The first thing it teaches us is that these bioelectrical patterns are in fact instructive. It's not just that, oh, I messed up the pattern and now the face is screwed up. It's not that. It's that you can actually induce very targeted, coherent changes. So we know it's instructive. It carries information. Make an eye here. That's the first thing. The second thing is the pattern that we put in is extremely simple. It doesn't have all the information that you would need in order to build an eye from scratch. If somebody wanted to really, let's say, 3D print an eye, nobody's done that. Not even sure it's possible. But if somebody wanted to do that, you would have to have an incredible amount of information. We didn't pass all that information on. So the second thing you learn from this experiment is that what we're dealing with is an extremely competent material. This isn't a passive material where you have to, as an engineer, you have to specify everything it's ever going to do. All we did was it's like a subroutine call. All we have to say is build an eye here. All the stuff that's downstream of that, all the gene expression, all the cell movements, everything else it happens, and then it stops when the eye is built. We don't have to tell it to stop.
And that could have been any cell. Any cell in the embryo could have been used for that.
I think so. We haven't tried. So we haven't tried every possible cell, obviously, but we did try. But we've seen eyes in pretty much every location you can think of. So yeah, it's pretty much everywhere. So that's the second thing you learn. The third thing you learn from this is something very interesting and I think profound, which is that if you look at the developmental biology textbook, it'll say that only the exterior tissues at the front of the head are competent to become an eye. And the reason they say that is because they have been using something called a master eye gene, this thing called PAX6. And this master eye gene, which they thought was the sort of top level thing that induces eyes, they would try injecting that into different parts of the embryo, and it never produces eyes except in the very front of the head, kind of roughly where the eyes normally form anyway. So they would say, so using that test, they would say, okay, fine, the other tissues simply cannot make eyes. So this tells you something interesting. When you estimate the competency or the intelligence of some other system, you're basically taking an IQ test yourself, because what you're really saying is, this is the best I can do in prompting that system. This is what I think it can do. This is my extent of understanding of how to communicate with it. And if somebody else comes along and says, you missed this whole set of competencies, and there's a way to trigger that, well, then you have to revise your perspective. So that reminds us to be very humble when we look at, and we have many other examples of this. When we look at a system, we say, well, it's a simple deterministic system. We understand the rules, so we know what it'll do. No, you absolutely don't know what it'll do until you check. And even then, you can't be sure that somebody else isn't going to find a better lens, a better way to look at that system and find out that actually you missed the boat. It actually does all this other stuff. So that's a good reminder for us to realize that these competencies are often say more about us and our understanding than they do about the system itself. And so, okay, so those are kind of the three things we learned. And actually, there's a cool one that's a fourth one, which is this: if you only inject a few cells, there's not enough of them to make an eye. But what they do, and they do this on their own, we didn't have to tell them how to do this. They will signal to their neighbors who themselves were not injected with anything, and convince them to work with them to make a much bigger eye. And it's because we can tell which cells we injected and which cells were neighboring cells. So that's kind of what you see other in collective intelligence doing, like ants, where a couple of ants will come across something they can't move by themselves. What do? They recruit other members from the colony, right? Cells do exactly the same thing. So all of this stuff are competencies of the material that's already there. So that's one example.
Working at different hierarchies, and I want to come back to that, because that's going to be crucial to the way we see self and agency.
Yeah.
But just to clarify, because it is important for the sort of, shall we say, the old way of seeing things, can you clarify what is the DNA responsible for and what is the bioelectricity responsible, just so we can visualize the two systems?
Sure. I will use the analogy that probably everybody's familiar with, which is computer hardware and software. And I am not claiming that biological systems exist in the kind of linear architecture that we use today. That is, they're not the kind of computers we use today. That's clear. People really freak out when
Maybe
I
super advanced AI many years in the future, but not as they are today.
but certainly not. And so everybody, I don't need to. I'm not trying to say that today's computers are a good model of what's going on. But the one thing that that is really useful to think about is the distinction between hardware and software, and in particular the notion of reprogrammability. So here's what the DNA is doing, and this is really it's really shouldn't be a controversial point because we can read genomes now. We know what's in the DNA. The DNA doesn't directly say anything about any of your body features. The DNA specifies protein. It specifies the tiniest level of hardware that's available to your cells to do interesting things. That is not that's not a controversial point. We've known this for many decades. That is what the DNA actually encodes. So the DNA gives you the hardware of your cell. Now, what's interesting about certain kinds of hardware is that once it exists and once the juice is turned on, meaning metabolism is there to power the thing, then all kinds of interesting new behaviors become possible. So if somebody, if I showed you a recipe for, I give you a bag of electronic parts, and I give you some of these parts are sticky and they stick onto other parts, and you sort of everything gets put together. As soon as you turn on the power, you find out that it does something. And what does it do? You find out that it's a calculator. And it starts off with a default state, and maybe it says 0 on the little LCD, but you soon find out that actually that's not all it can do. And if I give it certain stimuli, meaning if I punch some of the buttons, why then it does other things. And then you find out that, let's say it does, let's say it computes the truth table of the AND function, it can calculate an AND or something like this. And then you ask yourself, okay, so where was this AND function? And so you're looking at this, you're looking at the parts list. And you're like, wow, it's not anywhere on this parts list. Like, where did it come from? And so I think we have to be really clear that the question of where do things come from, maybe being patterns of shape, patterns of behavior, doesn't have a super conventional explanation, which is, why it's written down here. I mean, in some cases it does, but many times what actually happens is a consequence of not just the material, not just the hardware, but also of the laws of computation, the laws of physics, the laws of mathematics, and that's those are the ingredients in your explanation of where it came from. It's not just the hardware. So the DNA absolutely must provide the hardware. So, for example, if your lens is going to have certain optical properties, you need to have the crystalline protein, and it needs to have certain properties so that your lens bends light in a particular way. That's the DNA. Can't do it. The DNA is essential for that kind of stuff. You absolutely need the hardware, but it's only a part of the explanation. And we know this from computer science. We know that, when you and I are using our laptop and you want to go from Microsoft Word to Photoshop, you don't get out your soldering iron and start rewiring the thing, right? And that's because we understand that this is a multi-scale device, that the hardware can do many things, and that it's much better to interact with it via this convenient interface that it exposes to us. Exactly the same situation here.
Okay, thank you, Michael, for that clarification. But for me, the really big question going forward, there's a lot of big questions I want to get to today, but this is the first big one, is where does the form actually come from in each of these cases of elaboration? Yeah. Not from natural selection of genes, then where from? And where is the electrical field shape information stored, and how's it passed on? You speak about mind-boggling experiments where you can manipulate the size of the cells radically, but the salamander's form at the end of the experiment is exactly the same size and shape. Where is that template actually memorized?
Yeah. So I should be clear. That salamander experiment was not me. That was actually done in the 40s. So that's a very old experiment. So that's been known but ignored for many decades. So look, I think you're right. You're asking about the most interesting part of this, and I think there are two pieces to this. One piece I can answer, and the other piece we don't know. The first question is, where is it encoded at any given moment in time? And this we can now answer because we can see it. So, for example, now we can visualize it. So, for example, in planaria, in these flatworms, when you cut the head and the tail off and you leave a middle fragment, it will regenerate, and it typically always makes one head, one tail. So you can ask this question: how does it know how many heads it's supposed to have? And you might think, well, it's in the DNA somehow, even though, of course, we've read the genome, doesn't say anything about heads and tails, but you might think that somehow it's nailed down in the genome. But if you actually look at the electrical patterns in that worm, you will see that there's a particular pattern that we have decoded to say one head, one tail. And how do we know that that's what it says? Because we can change it. Because if we go in and change that pattern to be duplicated so that it really says now two heads, that those fragments will make two-headed worms. Not only do they make two-headed worms, but those two-headed worms, if you keep cutting them in the future, will continue to be two-headed. That memory is stable. So once you've convinced the collective that actually a normal worm should have two heads, it keeps that information until you go back and change it to being one-headed. So the answer to the question of where is the number of heads stored, that we know. It isn't in the DNA. It is in the stable bioelectric pattern maintained by the tissue.
But what generates that pattern, and if it is the cells or the proteins, where in the protein is that memory encoded?
So okay, so this is the more difficult question, and I think we need to really make progress on this. We really have to redefine what we mean by where. I'm going to give you a simple example. If you've ever heard of what a Galton board is, so just imagine it's a vertical board like this, and you bang a bunch of nails into it, right? Just randomly bang a bunch of nails into it, and you take a bucket of marbles and you dump this bucket of marbles over this thing, and you could buy this thing as I think it's $15 on Amazon, and it's a toy, a toy that it's very instructive. So you flip this over, you flip the bucket over, the marbles go bang 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 bang, and then they settle into a pattern. And something amazing happens, which is that if you have enough marbles, they always give you this lovely bell curve, right? This famous Gaussian distribution. And so just imagine you're looking at this thing and you ask yourself, that's a beautiful curve. Where is this thing encoded? Well, let me see. Is it in the wood? Well, I don't see it in the wood. Is it in the distribution of nails? No, you can move the nails around. You still get a Gaussian pattern. Is it in the marbles? You sort of stare at the marbles. I don't see it there either. Where does it come from? So we need to make our peace with the fact that when we ask these questions about where does it come from, the answer isn't in the parts list of the things you worked with. Some of that answer is, and I think this is so. Here's my attempt to understand where it is. I think it comes from wherever the truths of mathematics live. So the
It sounds like a probability distribution of quantum mechanics, but before a collapse.
And it may well be. This is so that's a little bit beyond my pay grade, but maybe, right? So really, the distribution of prime numbers, right? Where does that live? The truths of number theory. Where do they live? If all of, as far as I understand, and maybe some physicists will correct me, but as far as I understand, if all the conditions at the Big Bang were completely different and the laws of physics were different, the universe were different, the truths of let's say number theory would still be what they are. All the prime numbers would still be however they are. So here's what I think is going on. I think that what evolution builds is problem-solving machines that are really good at exploiting these laws of physics and computation. I think they're free lunches in the physicist sense of the word free lunch. I literally think they're free lunches. Here's a really simplistic example. Suppose you're in a universe where there's a particular kind of triangle that has the highest fitness. That's the best organism is this like triangle. So imagine evolution, right? So a whole bunch of generations go by and you found the first angle, and then a bunch of generations more and you found the second angle. Well, guess what? You don't need to evolve the third angle. You know what it is. You already have it. You don't need to spend time evolving it. And so because the angles have to add up to 180 in flat space. And so what this is telling us is that this isn't some philosophical fairy tale about this Platonic space that has various kinds of truths in it. It's very practical. Evolution just saved a third of its effort in finding this thing, and that's why I call it a free lunch. Because a lot of these kinds of things, computation, there's things from mechanics, from adhesion, just a huge amount of things that evolution exploits, and that's what I think is going on. So it makes these machines that are instantiations of these free lunches that come to us from, I don't know, from wherever mathematics comes from.
But that field of a tadpole that has its specific signature is obviously going to be very different to the way another organism develops. The specific shape to that organism must be related to the cells and to the DNA and the RNA in those cells somehow. It just seems too bizarre that it's not in the cells of that particular organism in some sense.
The word "in" here is doing a lot of heavy lifting. You're right. You're right, of course, because
our intuition takes us towards a materialist approach, because that has been the last 100 years of science. We're looking for it in the biological substance itself.
Yeah, but isn't it funny that Plato and Pythagoras were onto this however many hundreds of years ago, right? Without having all these amazing examples of reprogrammability and so on. So I think it's not. I'm certainly not the first person to have these thoughts. I will just say that you're absolutely right in the sense the part here that is correct is that typically frog eggs make frogs and acorns make oak trees, right? So obviously there's some connection here, and there is. What happens is that evolution shapes initial conditions, meaning the properties of those ion channels, so that by default they will give rise to a particular electrical pattern and chemical pattern and biomechanical pattern when left to their own devices. So we know we have lots of devices that reliably, a simple electric circuit, if it's designed correctly, when you turn it on, it will establish some sort of default baseline state that you wanted it to establish. Let's say again, it's a calculator and it's got the little zero. Why does it reliably go to zero? Because we designed it, or evolution shaped it that way. But that zero isn't in any of the parts, right? It's just that the parts were specifically selected for exploiting the laws of computation and the laws of physics in just the right way. So absolutely, there's an important role for selection, for evolution, for the hardware, no doubt. There's a twist to this, though, which is that as we're now seeing, you can take that hardware and rearrange it, or in fact let it rearrange itself. We don't do as much of the work as the material does, and then it will exploit other kinds of patterns that, as far as we know, have never existed before. So what that tells us is that yes, evolution can sculpt hardware for specific purposes, but I don't think that purpose is to solve a specific problem. I think that purpose is to be a problem-solving machine. It's a sort of second-order intelligence. And I think that's what evolution gives us, because you can have frog eggs that give rise to frog embryos, or you can have frog eggs that give rise to cells that make xenobots, and they have different behaviors and different competencies.
Now, moving on, this all has huge implications for how we're going to do this biological engineering that you've been talking about in the future, both for health reasons, but also for biotech. So engineering and training these active biological materials, so living cells that start with their own goals and preferences and agency, is much more dynamic and flexible, if a little tricky and slippery, than traditional engineering of passive materials. So the focus moves from building structure from the bottom up to training and guiding plasticity from the top down.
Yeah.
Now, we are not used to thinking of cells as being, using your word, agential, having agency and preference. But even primitive cells do. And contrary to this long-term evolutionary idea, they can be repurposed really, really quickly, responding to the environment. They're harder to control than traditional passive engineering materials. I think you talk about, piles of cats rather than piles of dogs rather than piles of Lego, to give an example. They are harder to control than traditional passive materials, but potentially far more robust and flexible.
Yeah.
So help us unpack all of that. You've mentioned many of these points already. Tell us about your Xenobot experiments and how they've shown us that this repurposing is not only possible, but actually somehow built into our cellular intelligence.
Yeah. So I'll give you a few examples, but I want to separate the Xenobots out from this because they're too new. We haven't really done enough yet. We're doing all that stuff, but the published work to date doesn't allow us to say too much about what the competencies of the cells are yet. We'll get there, but they don't yet. So I'm going to use some other examples. Because I want to be really clear. All the things that I'm saying in terms of the competencies of the tissue and so on, these are not philosophical positions. And many people have like baseline commitments to what cells must be and what they can't be. I don't think any of that is possible. I think all of these are empirical questions. You have to do the experiment and find out. So specifically in the case of Xenobots, I mean, of course, I expect to find all these things, but we haven't actually shown that in the case of the Xenobot. So we'll set those aside we can talk about evolution and things like that. But I want to talk about a few other examples where I think it's perfectly clear what's happening in all of these cases. And people have debates all the time about, is something, and they use different terms, but is it cognitive? Is it sentient? Is it intelligent? How much does it have, and all that? Let's just remind ourselves what all these words are for. Here's what I think they're for, and I think that trying to keep these binary categories is completely untenable. What I think these words are for is to express protocol claims. In other words, what they're saying is, when you've named the level of intelligence of something, what you're really telling me is, here is a set of tools that you can use to interact with it optimally. So if you tell me something is a simple machine, what I hear is, well, that means I can't guilt it into doing anything. I can't reward it or punish it. I can't change any memories. I just need to rewire it. I got to get in there with a screwdriver and a hammer, and that's how we're going to do this. If you tell me that, well, this is a cybernetic a homeostat, now I know something different. I don't actually need to rewire it or change it, but I do need to understand how the set point is stored, like a thermostat in your house. So you don't really need to know how the whole thing works, but once you know it's a thermostat, then you need to know, well, here's how I control the set point. And by the way, once I change the set point, I can walk off and it will do the hard work of staying in the range, right? Somebody says to me, oh no, this is actually a reinforcement learning system, or it can do associative conditioning or whatever. Now I know, I don't need to know almost anything about how it works. What I need to know is the currency of reward and punishment. And I need to know what are the timescales and what are the ways I'm going to train this thing. And then somebody says, well, this is a complex metacognitive human. And then I know I don't even need to specifically do rewards and punishments. I can actually give you give you reasons for things. And then if my reasoning is good, you'll go off and do everything yourself. I can convince you to do all kinds of stuff or not. Right. And so what you're really finding out is that, are you using the tools of rewiring? Are you using the tools of cybernetics? Are you using the tools of behavior science? Or are you using the tools of psychoanalysis and I don't know, spiritual development or something? So all of these things, you've got to figure out where something is relative to this scale to be able to use the right tools. You can see you can go wrong easily in either direction. Right. And people talk about attributing too much. There's this teleophobia out there where people are, scientists are really leery of attributing too much agency to certain systems because they're afraid that we're going to slide too far to the right. I think it's just as likely to slide too far to the left and have both practical and also ethical lapses when you don't recognize the degree of cognition that you're dealing with. So
the most basic single cell moves away from something that's going to damage it and moves towards something it can eat. I mean, that's cognition.
Yeah. Absolutely. And if you don't think it is, I made a little chart for people to make it like really kind of simple, so you can't really hide. If you really don't think that's cognition, then you owe a story about what happens when you go from being that cell to being a human. So where did the cognition kick in? And there is no good story to tell there. And so you know that you're not dealing with a binary category, you're dealing with a scaling. And that we do have a story that is developing about how the scaling works. So all that is to say, when you're dealing with let's say regenerative medicine or kind of biomedicine in general, you have to ask yourself, do we know where cells and tissues are along this scale? Because if you assume they're on the left part of the scale, where your only possibility is rewiring the hardware, so this is DNA editing, CRISPR, pathway engineering, those kind of things, then that's one set of approaches. But if you're wrong and the cells are actually further along, you're leaving so much on the table, right? And so that's my claim, and that's what we've been trying to do for some time now, is to use the techniques appropriate to other disciplines, which are specifically designed to help you relate to systems of higher agency, to see if we can communicate with these collective intelligences, not try to micromanage them, not play them like a puppet, but actually try to take advantage of the agency they have and do what biology does, which is, I think that's what biology does all day long in your body, is the cells are constantly hacking each other to try to convince them to do things. And that we can take advantage of that too.
But you've got to tell us about the Xenobots, just because they're such a good example of that extreme adaptability. I mean, they are extremely tiny systems, and they completely repurpose themselves. I think you purposefully left them alone to adapt on their own at first, to be absolutely clear that you hadn't trained them or manipulated them in any way. You literally just left them to see what they would do. Tell us about these. I mean, I think Xenobots is almost a misnomer, because we start thinking that they're some kind of programmed robot, but these are just cells being repurposed completely by their environment.
Yeah. So, and by the way, there's another. I can't really say much about it because the peer-reviewed paper isn't out yet. But in the next few weeks, there will be something out that will be along these lines that is really going to nail some of these points even harder. But I'll just talk about
that. Will be in the show notes. So
yeah.
We'll reveal that via the show notes. Like
So the thing with Xenobots is basically, and this is the biology of all this, was done by Doug Blackiston in my group, and this is all in collaboration with Josh Bongard, and back then his student Sam Kriegman did some of the AI work. So the idea was simply this: what happened in traditional plants and animals when you ask? This is related to the question you were asking before about shapes. When you ask where did that shape come from, one way to answer that is to say, well, selection, right? So basically, you live in a froggy environment, so everything that isn't shaped the way you need to live in that froggy environment died out eons ago, and so now you're left with this. So there you go. So then we specifically wanted to ask this question, but could we use the exact same hardware to dial into patterns from that latent space of possible form and function to find new ones that were never here, that never were the subject of direct selection? Right? The cells, of course, were, but the organism wasn't. So we did something very simple. So Doug takes these early frog embryos, and he takes the ectoderm, which is the outer skin layer, off of the top of the animal, dissociates all the cells, puts them in a little pile in a petri dish. Now you might imagine there's first of all, this is in a weird way. This is a kind of model of life after death, in the sense, because the original embryo is gone. It's been disassembled. It's into pieces. So that's gone. But the cells are still alive. And so what happens next? So many things could have happened. The cells could have died. They could have crawled away from each other. They could have done absolutely nothing. They could have made a flat monolayer of like tissue culture. Instead, what they do is they come together and they form this little ball. And because the skin normally has these little hairs that are used to distribute mucus down the side of the frog. These little hairs can be used to actually swim against the water. So they start to swim, and so they swim, and they have their own little different patterns of motion. They can go in circles. They can go back and forth. They can do all sorts of things, and they have all kinds of other behaviors. We are just scratching the surface of this. So what we've seen so far is the motility. We've seen regeneration. So if you injure them, they will try to come back to their new Xenobot shape. They do this amazing thing called kinematic self-replication, which is basically von Neumann's dream of a robot that finds parts in its environment and puts together a copy of the robot. So if you sprinkle a bunch of loose skin cells, what they will do is they run around, and by their motion, they will compact these skin cells into little piles. And because they are dealing with an agential material, much like we were when we made the bots, guess what happens? These piles become the next generation of bots. And what do? They run around and make the next generation. Now, this is there's never been any Xenobots. There's never been any selection to be a good Xenobot. There's never been kinematic replication as far as we know, no other animal does this. And again, it's this like amazing thing where when one view of this is to look at that process and say, well, there's no magic here. I see how this works. They move in circles. That will collect cells. The cells are sticky. This and that. Of course, you can tell a physical story about what's happening. It's not going to be fairies that are responsible. It's always going to be physics and things like that. For sure, that's not magic, but the whole thing together is really interesting, because after we did it, you can say that you understand how it works, but no one knew that was going to happen. No one could have predicted that was going to happen. No one did predict. Much like with most of the all the bioelectricity stuff that we do, like after the fact, you can see what's going on once we show the mechanism, but no one saw that coming. And so that already tells you that we don't have a real good science of understanding what these novel collectives are going to be able to do. Now we specifically did not engineer these cells. We didn't put in circuits like synthetic biology. We didn't use any weird nanomaterials. We didn't inject them with any drugs. We didn't edit the genome. If you sequence the genome, all you ever see is Xenopus laevis, because we didn't put any transgenes in for the first 3 pages. We will do all that later, but we didn't do that to start off with, because I didn't want people to say, well, you've engineered the heck out of these cells. No, you did that. Now, Doug worked very hard to sculpt them. He's this amazing microsurgeon that gets the skin cells off and everything. But we did not design kinematic self-replication, right? The material does all that.
This marriage of cellular plasticity and bioelectricity is particularly important for the way we approach regenerative medicine and aging research, because if we can train cells naturally flexible plastic ability to repurpose depending on what's needed at the biological level, rather than having to use sort of corrective medicine that treats symptoms and not causes, or edits complex genomes, we can crack some of the biggest unanswered questions in medicine and biology, basically how to regenerate tissue and how to extend life. So let's come back to planaria, Michael. Describe these remarkable creatures that you studied and tell us how that their regenerative abilities, the fact that they don't age, and this particular way that they inherit mutations can shed some light on how one day we might be able to apply this regenerative ability of cells to ourselves.
Yeah. Well, this gets back to your question about the genome and what's actually in the genome. So when I was going through the biology classes that I had, and I assume nowadays everybody else gets the same thing, is that they tell you that the genome is really critical because it determines the structure and function of your body. That's the story you tell. So from that, you would assume that the creatures with the most precise control over their genome would be the ones that had the most robust and stable anatomy. Now, let's consider these planarians. So in these flatworms, what happens is that, first of all, they have a few properties. They are highly regenerative. So if you cut them into pieces, and I think the record is like 276 pieces or something like that. So you cut them into pieces, each piece will grow into a perfect little worm: head, tail, brain, eyes, intestine, the whole thing. So super regenerative, very high fidelity. So they rarely make mistakes. They're very, very, very high fidelity. They are cancer resistant. Okay, so they're very resistant to carcinogenic transformation, and they appear to be immortal. In other words, no one's ever seen an old asexual worm. The ones there are species that in fact seem to age, but the ones that we work with are asexual, meaning that the way they reproduce is a single worm tears itself in half, each half regenerates the other half, and now you got 2 worms. That's how they reproduce. So those seem to be immortal. They don't age. There's no sign of aging there. So, but there is one weird thing, which is that if you reproduce this way, meaning that both parts of the body go on to form the next organism, it means that any mutation that you suffered in your lifetime that didn't kill the stem cell that it was in is going to amplify as that cell gives rise to the other tissue needed to regenerate you, right? So unlike us, the thing with us and most organisms is that if you get a mutation in your body, or if you exercise and build muscle, or if you whatever it is that you do to your body, the embryos don't inherit that because they come from an egg, not from the rest of the body. In planaria, that's not true. The next generation literally comes from the whole rest of the body, which means that it just accumulates mutation. 400 million years, their genome is a mess. Their genome is a total mess because of it, because they keep all these mutations, right? And so now it's a bit of a scandal. The animal with the best cancer resistance, immortality, and regenerative ability has this incredibly messy genome. What's going on here, right? That shouldn't be that. It should be the opposite. It should be the other way around. This drove me crazy for a couple of decades now, but I think we finally have a bit of a handle on what's going on here, and it really tells you that so we've created, for example, worms with 2 heads. They are permanently 2-headed. If you sequence the genome, you are none the wiser about the fact that they're two-headed. There's nothing wrong with the genome. You have no idea that they're two-headed from looking at the genome, right? So this tells you there's a real gulf between the genomic information and the actual structure and function.
Mm. And that really is telling, isn't it, about the conversation from before about where is that being held? Because if it's not in the genome, if it's not in the protein, that inheritance is very, very short term. Very, very interesting. Now, in order to make regeneration a priority, which obviously is your end goal, is to do this for humans, to make it a priority for a few cells, we need to understand how a collection of cells, each with their own minimal goals and agency, knows that it's become part of a single larger collective and then combines its goals and objectives into a single self with these sort of hierarchical competence structures, they're called, aren't they? These hierarchies of bigger and bigger objectives, and how the agency and the objectives then get more and more advanced at each level of complexity. And to explain this, we need to make a last point on bioelectricity, which is how collections of cells communicate using electrical signals, something you mentioned earlier, which we're used to in brains, but we may not imagine as so fundamental in cell communication. Explain to us Werner Lowenstein's extraordinary discovery of gap junctions that permit cells to share these electrical signals and recognize each other as part of the same organism and collective rather than the outside world, and how that sort of builds up to having this sort of cognitive self.
Yeah. So the mechanics of it are that these gap junctions are little tiny proteins on the surface of a cell that make almost like a submarine hatch where two of them can dock like this. And when they dock, then small molecules, not just electrical current, but all kinds of like very small ions and other small molecules can go back and forth. Not proteins, not DNA, but small signaling molecules. And what we are trying to understand, and this is very much work in development, so we do not know the full answer to this, is when you make a network out of cells that are connected that way, you gain certain remarkable abilities. The ability of that network to store memories, to pursue goals, scales up. This is a little bit parallel to, the architecture is quite different, but it's a little bit like what we're learning now in AI, that when you put together large networks of simple subunits, you gain functionality that really was not obvious at all from looking at the parts. Right? There's a lot of emergent capacity that comes up. So I'm going to give you a very simple but potential story for why gap junctions are kind of magical for scaling up these collectives. Imagine that you've got two cells, A and B, and the canonical type of signaling is that cell A lets out some sort of chemical. The chemical diffuses, comes over to hit cell B. Cell B knows for sure that came from outside. So now cell B has some options. You can ignore it. You can do something about it. Maybe it's information that you need. It's a signal that comes from outside, and you know that. And whatever the information is carried by that signal, you also know that that's not your information. That's the information about something else. But now imagine these two cells are connected with these gap junctions. The events taking place in cell A pretty much immediately get communicated to cell B, but they don't go through the outer surface. They appear in the internal milieu of cell B. So let's imagine for a minute that cell A gets damaged. Something pokes it, and there's a calcium spike, and that calcium spike propagates to the next cell, cell B. Cell B has cell B has a calcium spike. Calcium doesn't come with little labels that says the ownership metadata. Whose calcium were you? All you know is that you've got a calcium spike that's indicative of damage. So what it really is a false memory, but you don't know it's a false memory because it's your memory. It's a trace of an experience that you know how to interpret, but you have no idea. You don't have and this is actually, I actually think this is really profound on a philosophical level. It reminds us that we don't have access to the past. All we have access to are the physical engrams that the past has left in our body and brain, and we interpret those at any given moment. You reconstruct yourself from the evidence that the previous selves have left for you. You don't actually know anything about what happened in the past. You have to reconstruct it actively. So the cell gets this signal, and now what you find is that because of these gap junctions, the ownership of memories gets fuzzy. So instead of your memories and my memories, when the memories are separate, we can remain individual. We can remain as individuals, and that means that we can have our own goals and we can compete and we can do all kinds of things because we are individuals. Once you are connected informationally, your memories are my memories. When that happens, and they may not all be, not everything goes through gap junction, so it's a partial kind of mind meld. It's not a complete mind meld, but we have this mind meld enough so that once we start sharing our memories, meaning we share a view of the world, we share a reconstruction of what happened, what's likely to happen, and so on, we become one larger being. We have more computational capacity. We're bigger. We're physically bigger, which means, if you have lots of cells, now you can measure things that happen at quite a distance away. You're not just measuring like single cell local states. And so what you're scaling up, and it's a large coherent being, because you can't compete with or defect against yourself. There's no point in doing anything bad to the cell next to you because it comes back to you immediately, and you can't even have those thoughts because all the memories are shared, right? And so it really is a mind meld, this like collection. So I think what's really great about gap junctions is precisely this ability to generate a melding of memories, which then give rise to it. It's not you and me, it's we after that. And that's it.
And the cancer cells is relevant here, isn't it? Because actually one of the first things we see with cancer is the gap junction closing. So the body assumes that's an outside thing. And the cancer cell has absolutely no allegiance to the cells around it.
Not only doesn't it have allegiance, so I want to be clear about that. The cancer cells. So sometimes when people do like game theory models of cancer and so on, they kind of treat those cells as more selfish in a way. I have a different view of this. I don't think the cancer cells are more selfish at all. They have smaller selves. So the other cells are not individual amoebas. They are a liver or a piece of skin or some other organ. They are just as selfish. It's just their self is huge, right? And so that computational connectivity, not just the physical boundaries. I don't mean the physical boundaries of where the thing is. I mean and we call this a cognitive light cone. The size of the biggest goals that you can pursue in space. When you're a single cell, your cognitive light cone has shrunk back to amoeba size. And then, of course, as far as you're concerned, the rest of the body is just environment to you, right? It's just external environment. And so then, yeah, you get metastasis. Amoebas go where they want. They eat what they want because they're in an environment.
So each step up on the hierarchy of collective agency would feel like it was a cognitive self with its own agency, its own objectives, working towards those goals, right? So to what extent is that a real self rather than just the impression of being one? Because presumably there are little mini selves at every stage of those objectives scaling up. And the big one is the one that particular collection of cells would experience. To what extent is there a real self rather than just the impression of being one? Because that has implications for personal experience of consciousness and agency, right? Free will.
It does, yeah, absolutely it does. So I want to say two things that may initially sound opposite, but I think they're both true. On the one hand, I think they have exactly the same reality status as the not in size or scale, but ontologically they have the same status as the cognition that you and I have. In other words, they operate in different spaces. I think your liver operates in a physiological state space and solves problems and has memories and so on. But I think there is no reason to think that those selves, even though we have no access to them, have any less of an inner perspective than we do. I mean, it might be smaller and it might be more simplified and whatever, but it's just as real.
So to clarify, they may be having some kind of subjective experience, those smaller.
If you and I have subjective experience, so do they. There's no principled way to say why they don't. Now, some people think that you and I don't either, right? There are eliminativists about all of this, but I am assuming we're not going to.
Is there a way of testing that? Like what it's like to be a liver, or?
Well, sort of. There really isn't a way of testing that for you and I either. I mean, these are deep waters. Very smart people have argued about this for a long time.
This is why I've steered us here before while we still had time, because this is where my most important questions lie.
I'm looking at the background behind you. I'm thinking we're gonna get to this sooner or later. So, so here's the thing. People, when I say, look, all of the other components in your body are doing all of the same things that your brain does at different time scales, with different degrees of complexity in different spaces. They don't all talk. They don't all live in linguistic space, but they are doing the same kinds of things that your brain does, and therefore, if you think the brains are in some way sufficient for consciousness, then so do you have to take seriously that these other things do as well. In fact, I've had a lot of arguments with people who develop specific theories of consciousness, and I would say to them, so what is it about brains that's really special? And I say, well, brains, have these features. Okay, every tissue in the body does this, and so I have yet to hear anything that's quite specific to brains. But then what people say is, well, I don't feel like my liver is conscious. That's a mistake. You don't feel like I'm conscious either. So you, meaning your left hemisphere and maybe some other stuff, feels like it feels, but I haven't heard from your right hemisphere yet, and I definitely haven't heard from your liver. And whatever consciousness you have, you infer my consciousness. And if you were better at understanding behavior, and why do you infer my consciousness? From my behavior in three-dimensional space and linguistic space. And if you were better able to visualize physiological space, transcriptional space, and anatomical space, you would use exactly the same evidence to say, wow, that liver, not human level cognition, but it's definitely got something that it's like to be solving those problems and to be struggling for its life and in the face of all these stressors and so on. So my first answer is, I think they are absolutely as real. Your body is full of selves that have as real, if not the same scale or the same kind of experience as you do. That's the first thing. But now I'm gonna stand the whole thing on its head and say this. The other choice you gave me. So you said, is it real or is it just a perception of the consciousness, right? So I'm gonna go now. Really lean into the second thing, which is this. I think if we really ask ourselves what selves are, I really think that what we mean by real is just this. They are a useful model made by an observer. Now, that observer is not just an external observer, it's also the system itself. You observe yourself and make a model of yourself as an agent, which I think is really critical. But so the bottom line is, I don't think these two things are a dichotomy. Like I don't think they contradict each other. I think
they're just useful schema, useful representations.
It is a useful, and it is maybe the most useful of all. So people who say it's an illusion, I would say that, well, it is to the extent that everything is you making a representation of cutting up your world in some way that you can understand it. So from that perspective, much like, the tables and chairs and computers and the economy and all this other stuff, all of it is an illusion in a certain important sense, because this is our attempt to make sense of our world. But in another sense, they're as real as anything, because it's maybe the most useful illusion there is. So the illusion that you are, and I understand there are many people working on breaking through that illusion in terms of Buddhists and meditation and all that. Like I understand all that, but just for the purposes of being in the world, right? Not seeking to escape embodiment, but being in the world requires you to have a model of yourself being an agent that does things, which necessarily means having a demarcation between you and the outside world, and a list of things that you think you actually have control over. So we get to the will idea. So both, right? Both. It is an illusion, but it's one of the best ones we have, one of the key ones that we can develop.
So all of this is building up to the big question that really is the reason I've invited you on the show, Michael. I just really got to get to this. So we've built it up, these various hierarchies of more and more complex selves with more and more complex objectives and agency. So my big question, and I'm speaking to John Vervaeke about this later in the series on collective intelligence, the physical science of how our ability to solve problems as a collective intelligence works, it just isn't clear to me. You mentioned ants there, that these are separate organisms working together in a very, very clear way. If your model of hierarchical selves is true, then what's to say that the cells of our various bodies coming together to solve problems collectively couldn't be seen biologically as a collective self? Or perhaps even all the cells of this planet could be seen as a collective self, as with the goals and the agency in Gaia theory, or even the sun as a self, as proposed by Rupert Sheldrake, or pushing even further still, cosmic intelligence as experienced in mystical experiences, the entire universe as a single self, each at a higher level of hierarchical competence with higher level goals and agency. How do we draw the line between connected and aligned systems of cells unified by electrical fields and signals and other types of interconnected and collective intelligence?
Great question. So I'm not sure of anything there, but I do believe one thing very strongly, that these things are to be decided by experiment. So it is not the case, I think, that you can take any old thing and say, I think there's a high level self and somebody else, let's say it's a city or a town, and you say, I think there's a high level self there, and somebody else says, no, there isn't, and forever they're sort of left to argue, and each remains in his own opinion. I don't believe that's where we are. I think these are empirical questions. So how do you decide that something is an actual agent, or better yet, because I don't believe in binary categories, to what extent is something usefully modeled as an agent? So what I think we are developing in my lab and a bunch of other people is a set of tools to formalize this notion and be able to do empirical experiments. So let's here's how I see this. Any hypothesis of agency has to have a couple of ingredients. When I bring you a system, and this may be a paramecium, and it may be some weird synthetic biology that I've made, or it may be a whole country, or it may be the whole universe, whatever it is. I bring you a system, and here's what you've got to do. You have to make a claim about what problem space you think it's operating in. And it doesn't have to be the three-dimensional space. It could be a really weird space. It could be financial space. It could be linguistic space. It could be transcriptional space. There's some problem space that it's working in. You are going to make a claim about the cognitive light cone that it has, meaning what do you think is a useful size of goals that you think the system is able to pursue. And you're going to make some claim about how much competency does it have to pursue those claims. Is it like a thermostat? Is it like a self-driving vehicle? Is it like a dog? Is it like a human? Is it a bodhisattva? What is it? So all of those things are empirical claims. So person A says, "Oh, I see this thing pursuing goals in this kind of space." Person B says, "Oh, you're crazy. It's just a pile of it doesn't scale up. It's like a pile of rocks is no smarter than any individual molecule. It's just it's just a linear sum. There's nothing here." The way you decide is with perturbative experiments. So this is really important. You cannot look at behavior of any kind with a pure read, a pure non-destructive observation and conclude anything. People do it all the time. They look at something and go, look how smart this is. And this is why this is, as much as, I love the Xenobots. I'm sure they're doing smart things, but we have to be really clear. When people look at them doing all these things, they watch a video and they go, oh, that Xenobot clearly wants to go from here to them. You cannot conclude anything from a purely observational video. And William James had all this nailed down a long time ago. The way you test these claims is you do perturbations. You figure out what you think the goal is, you put a barrier in their path, and you see what competencies they have to get around it. Then you can start making some claims, and you can say, well, this is about the level of a random walker, or this is something that has a little bit of memory, or this is something that can plan forward 10 steps into the future. Those are all experimental results. You cannot derive any of it just from observations. You have to make a hypothesis about what the goals are. You have to, in whatever space, then you perturb it by giving it new challenges, and then you see what you've got. So for some systems, we've been doing this in behavioral science. We've been doing this forever for all kinds of systems. The behaviorists are actually pretty good at this for all the limitations. But then for other systems, it's really hard. And so here you run into these, but it's not impossible. And here you run, like somebody said to me once, your view, somebody said, well, your view is basically panpsychism in the sense that this is spread throughout the world. And I said, but potentially, yeah. And they said, well, how about the weather then? I suppose you're going to say the weather has hopes and dreams. And I said, well, be careful, because has anybody tried training the weather? We have not tried. We don't know. We don't know if some particular, for example, a pattern of, I'm totally making this up. I'm making no claims about tornadoes and whatever, but it is not clear to me that those kind of patterns don't have some sort of habituation, some sort of sensitization, some sort of associative learning. And as a simple example, people looked at gene regulatory networks, which are these very simple deterministic models of, got 6 or 7 genes and they sort of turn each other on and off. And you look at it and go, well, that's such a simple system. Surely that's going to be, dumb and mechanical. It turns out, and Richard Watson was the first person to find this, and we did a bunch of work on it. Turns out they can do 6 different kinds of memory. Just like that, with nothing new, no magic, no indeterminism. They can do 6 different kinds of memory, including Pavlovian conditioning, which actually has huge implications for the way we use drugs in medicine. So you cannot tell what you're looking at by pure observation. You make hypotheses and then you test them. And so that's why I think I'm fine with anybody saying Gaia, ecosystems, planet, great. Make the hypothesis and let's figure out what space you think it's working in, what perturbative experiments we can do, and then we're going to find out. And if somebody has a hypothesis that no, it's actually much smarter or much less intelligent, we can find out. That's the one thing I insist on, is that I really think these are all empirical claims.
Just to take a sort of easy example, you mentioned ants, right? Those are many organisms. They're beyond a single organism. How does their collective intelligence work so brilliant? Are they a single self? Is there an underlying mechanism that, as we've found out in this discussion, is very, very hard to locate, where their collective memory, where their ability to move as one it resides? Is it possible that beyond actual individual organism's body, there is a sort of guiding principle, a collective intelligence principle, where collaboration does come together in this unified way?
Yeah, I mean, I'm not an expert on ants, but from all the work that I've seen, it seems extremely likely to me. I mean, ant and termite colonies, they regenerate. If you damage a part, it will get rebuilt, right? And you can do a million. There's this great book from I think the 1920s called "The Soul of the White Ant" by Eugene Murray. It's very old, and he talks a lot about this stuff, about what the collective behavior of the hive or the colony is in these various spaces. And so it all boils down to this: you've got person A that is going to use a lens of some level of collective intelligence. You've got person B who doesn't believe it and is not going to use that lens. And now it's a very simple question: who does better in terms of being able to predict, control, and relate to that system? I mean, these are very empirical questions. You pick your lens and you see how you do. And that's true for regenerative medicine. It's true for all of these things.
Well, listeners, we're going to be going into depth with this, with John Vervaeke talking about this. But is there anyone, Michael, in the biological field working on collective intelligence that we should be looking into, as far as you know?
There are many. There's a page on my website, which we can add to the show notes later, that lists some of my favorite labs. I mean, there's some amazing people out there doing some of this work. And of course, I should say my list, lest I leave anybody off my list, is hardly exhaustive. But it's some great labs to watch in this space. There's a lot of work. It's a field of diverse intelligence. It's blowing up. It's the most exciting thing.
I definitely want to get some more biologists on the show to talk about how these collectives come together in such a unified way. Now, last point before we say goodbye, Michael, I know you've got to go. You mentioned Lowenstein, who discovered these all-important gap junctions that ratify a signal from the other cell as aligned and therefore to be sort of collaborated with as a self. You mentioned he was writing a book about consciousness when he died. What do you think the book would have been about? And if you had to finish it, what would you argue following his research?
Wow, boy, that's what a question. Well, I mean, so I think Lowenstein was a genius. Far be it from me to try to guess what was going to be in it. I will say that I met him. I think it might have been 2006. I drove down to Woods Hole and I talked to him, and he told me that he was writing this book. I don't know what's in his book, but I will say that. In my head, I have kind of 3 books rattling around in my head, which I don't, the first one I'm committed to write. It's a book on bioelectricity. So that one I'm actually writing. The second one.
Come back on when it's finished, please, Michael.
Yeah, of course. So One Pagan and I together are writing it. He's a neuroscientist. So that one's definitely going to come. And then the one after that, if I ever get to doing it, is really going to be on this issue of the origin and scaling of cells through biology and beyond. So
gap junctions.
Sure, yeah.
Oh, Michael, there is so much more from your work I wanted to get into: unconventional cognition, distributed cognition, the implications of bioelectricity for the nature of reality, bioengineering versus mechanical engineering, the implications for AI, if it could fuse with this biotech. But I think we have covered so much given the time that we had. So, as I said, if you do finish this book, please come back on the show and tell us more about these deep philosophical implications.
Yeah, thank you so much. I'll absolutely come back. Yeah, there'll be in the next couple of months. There's a few stories, probably 3 or 4, pretty cool stories that are going to come out. So I'm happy to come on anytime.
So it just leaves me to thank you sincerely for making time for us,
so
and to ask you to keep up this amazing research that is constantly overturning all of these things in the consensus. Michael Levin, thank you.
Thank you so much. Thank you. Anytime.